Hello children, today we will be dealing with class 12 biology chapter 3 human reproduction part 2. In part 1 we studied the male and the female reproductive organs and how they are placed and what are the organs associated with human reproduction. As we have earlier discussed that the sexual reproduction involve three major processes: pre-fertilization, fertilization and post-fertilization events. So this is a pre-fertilization event and which has the first part, that is gametogenesis. Gametogenesis in human being is either the male gamete or the female gamete, and both the gametes are produced by different animals and different organs involved in this. So if you move to the process of gametogenesis, it can be defined as the process where the primary sex organs, the testes in males and the ovaries in the female, produce gametes, that is sperm and ovum. We are also seeing that the production of gamete is a very intricate process and it involves various hormones, endocrine glands and other important body processes in it. So we'll take the first process. Before taking the first process, that is gametogenesis or spermatogenesis, we'll again try to revise. As in the chapter 1 we have seen that the gametes are produced by the meiocytes. And what are meiocytes? These are the cells that will undergo meiosis. So in human male and female reproductive system, the meiocytes are present and they will move and they will form the 4 haploid individuals. Okay. So from diploid parent to haploid individual or haploid progeny cells. The meiosis take place in 2 steps, that is meiosis 1 and meiosis 2. Gametogenesis in human being is either a spermatogenesis or oogenesis. Spermatogenesis in male and oogenesis in female. So starting with spermatogenesis, as we have already discussed in the earlier lecture, that the spermatogenesis was taking place in the seminiferous tubule and the special arrangement in the testis is there, which is involved in the formation of sperm. So in this unit or in this part, we'll study how the process is taking place. The spermatogenesis start in the testis or seminiferous tubule. The wall of seminiferous tubule bears large number of spermatogonia which are diploid. These spermatogonia, they divide and they increase in the number. As they increase in the number, they also undergo, some of them, they undergo differentiation and they start doing meiosis or meiotic division. The first meiotic division start, and by the first meiotic division there is a formation of secondary spermatocyte. Okay. The primary spermatocytes were the special differentiated type of cells that are ready to undergo meiosis, or we can call them as a meiocyte cells. Now after formation of two secondary spermatocytes, again the meiosis continues and meiosis 2 occurs. The meiosis 2 makes the four different spermatids, and because these spermatids are like normal cells, they are oval in shape, but now they are going to be differentiated because they have to perform a special task, and the differentiation take place and they change from spermatids to spermatozoa, which are nothing but they are the sperm, and this process is called as spermiogenesis. We can understand from this what is spermiogenesis. It is a change of the spermatids into spermatozoa. After the formation of spermatozoa, this spermatozoa has head, neck and tail. But to provide or to get energy, they want nutrition, and they get attached to the Sertoli cells so that they get proper energy for the movement, and this process is called as spermiation. The spermiation involves attachment of sperm with the Sertoli cell, and now the sperms are ready to get involved in the process of spermatogenesis or the fertilization processes. This is also one diagram that clearly shows how spermatogenesis and where spermatogenesis is taking place. If you see, this is a testis and the scrotum, then we have the seminiferous tubule. When we see the cross section, we see the first, all the cell, they are diploid cell. Some of them they differentiated and they form primary spermatocyte. Then meiosis 1 occur, there is a formation of secondary spermatocyte. The secondary spermatocyte undergo meiosis 2 and there is a formation of N or haploid cells from a 2N or diploid cell which has 46 chromosome, and now we have 23 chromosome containing 4 cell. Now if you see this, they change from a normal cell to the differentiated sperm cells. Now if you see this, all the things are under the control of hormones. The glands associated with the spermatogenesis is the first most important gland is, or the area of secretion is hypothalamus. Hypothalamus release GnRH, that is gonadotropin releasing hormone, which stimulate the anterior pituitary, and the anterior pituitary release luteinizing hormone and FSH or follicle stimulating hormone. LH acts on the Leydig cells, and if you remember the Leydig cells, they were placed in between the seminiferous tubules, or they are also called as interstitial cells. They start synthesizing androgen, especially the hormone testosterone, which is involved in the formation of or the process of spermatogenesis. So we have another hormone, that is FSH, follicle stimulating hormone, which acts on Sertoli cell and it secretes some factors. These factors they help in the process of spermiogenesis or the differentiation of spermatocytes into spermatozoa. Okay. So these are the hormones that are involved in the process of spermatogenesis. So the final product of spermatogenesis is a sperm. You can see the sperm has three different the head, the neck, and the tail. The head, this is a complete cell, but this is a differentiated form of a cell. So what is this? It is covered by, just like some other cells, they are covered by plasma membrane, and we have this. The sperm has acrosome, which contain the enzymes that are involved in the formation, in the process of fertilization. Without acrosome, or without the enzyme presence in acrosome, there will be no fertilization taking place, because to degrade the ovum layer, during fertilization, the enzymes, they are very, very important. Then it has a nucleus, haploid nucleus, which contain the chromosome material. Then the neck has middle piece and the mitochondria. The mitochondria is very important because it will provide energy for the movement of this tail. So the tail movement is because of the energy provided by mitochondria. So this is a cell, but differentiated cell, or we can call it as a sperm. The human male ejaculates about 200 to 300 million sperms during a coitus, of which for normal fertility 60% sperm must have normal shape and size. This you will read in the next chapter. Some of the couple, they don't have child or children, so the causes of not bearing child can be with the sperm also. The motility, the size, and the numbers, they also determine the process of fertilization in normal human males. So this is all about the process that we call spermatogenesis. The another process involved in the gametogenesis process, which take place in a female and especially in the ovary, is the process called oogenesis, or the formation of ovum. Oogenesis is initiated during the embryonic development stage. Now this is very important. It is initiated. It starts at the embryonic stage and it occurs in the ovaries. It begins prior to birth. Before the birth, the process of oogenesis starts. This is a structure, or this is a flow chart that shows how oogenesis involves the process, or what are the different stages of life of a human female, where the process of oogenesis take place. Now you will see the first is oogonia. Oogonias, they are the very normal cell that will undergo the differentiation and they will change themselves into primary oocyte. So oogonia, there are large number of oogonia. They will undergo mitosis and increase in number, and then they will become primary oocyte. Now these all will be diploid. The primary oocyte will undergo first meiotic division. When it will first meiotic division? Before the birth, yani during fetal life. Okay, the first division starts, and this division is completed prior to ovulation. Prior to ovulation means the first meiosis 1 start with a birth, but it completes at the end of puberty. As the puberty starts, the meiotic division completes and there is a formation of secondary oocyte. This secondary oocyte and a polar body. So this is because of meiosis 1. The first polar body has smaller in size because the secondary oocyte will carry all the cytoplasm to preserve the cytoplasm, because ovum has to perform a very essential task. That's why this could be the reason for the less amount of, or the small small size of the first polar body. During the adult life, the meiosis 2 starts, but it does not complete, and it completes only at the time of fertilization. So we can call this meiosis as the longest time taking process of meiosis. Now the events are taking place in a ovary. So ovary is involved completely in this formation of ovum. So how they are involved? So this is, each primary oocyte gets surrounded by a layer of granulosa cells, and these granulosa cells are now called as what? Primary follicle. Here you can say this is a primary follicle, and primary follicle has the primary oocyte in it. The oogonium start the process of meiosis and get arrested at this stage in prophase 1. So up to prophase 1 it get arrested. These cells then originate from oogonia that are arrested at the prophase 1. They are called as primary oocyte. Then the large primary follicles get degenerated between birth and puberty. At puberty, therefore, there are only 60,000 to 80,000 primary follicles in the ovary. So the primary follicles, they get degenerated, and very few of them they remains. And those who remain, they change themselves and they become what? Secondary follicle. How they become secondary follicle? They attain one more layer. This layer makes them as a secondary follicle. After the secondary follicle, they change into tertiary follicle by this cavity, by generating this cavity which is called as antrum, and the walls, they become more and more thicker. There will be two layers now, thicca interna and thicca externa. Now here, what is there inside this? This is a secondary oocyte, and there is a formation of first polar body. The secondary oocyte retains much of nutrient as we have discussed, and one more layer, that is called zona pellucida, is formed around it. Now the Graafian follicle now ruptures to release the secondary oocyte. Graafian follicle is the final form of, or the mature form of follicle that is ready for ovulation, and the ovulation take place and there is a release of ovum, but the cell division is not yet complete. If fertilization take place, or the sperm penetrates secondary oocyte, then it trigger meiosis 2, and the meiosis 2 starts. Oocyte nucleus divides, one nucleus stays, combines with sperm nucleus, and the other nucleus leaves. The other nucleus means the polar body. Thank you.